Cells are the major building blocks of all living organisms. In order for an organism to grow, cells need to multiply, and this happens by a process called cell division. There are two different types of cell division in humans. Most cells divide by a process called mitosis, in which one mother cell gives rise to two daughter cells. In contrast, the process by which our sperm and egg cells are produced is called meiosis, and it involves one mother cell undergoing two successive cell divisions. The critical difference between mitosis and meiosis is the amount of genomic material, or DNA, that the resulting daughter cells end up with. Healthy human cells contain 46 chromosomes. These are not unique, but are comprised of 23 pairs, where one chromosome of the pair is from each the mother and the father. The pairs are called homologous chromosomes. When cells are ready to perform mitosis or meiosis, all chromosomes have been copied so that each individual chromosome is in turn composed of two identical sister chromatids. During a mitotic cell division, it is these sister chromatids that are separated, such that the resulting daughter cells continue to have 46 chromosomes. Conversely, during the first meiotic division, cells will actually segregate their homologous chromosomes, thus halving the chromosome number from 46 to 23. During the second meiotic division, which is similar to mitosis, sister chromatids are separated. Therefore, the first meiotic division is specialized, and the cell division machinery needs to be adjusted accordingly. A central element of the cell division machinery are specific enzymes called kinases. The function of these enzymes is to modify proteins within the cell with a chemical group called a phosphate group. This modification can change the function of cellular proteins, for example, by activating proteins that were previously inactive, or by relocating proteins to different parts of the cell. My name is Stefan Gallagher, and together with my former supervisor Dale Marston at the University of Edinburgh, I've recently written a review in BioEssays about a group of proteins that we think modifies the function of kinases during the first meiotic division. These proteins, which we refer to as Meiosis I kinase regulators, or MOKIRs, are specifically expressed during meiosis. We believe that it is the modification of kinases by MOKIRs that brings about the specialized chromosome segregation pattern that leads to the halving of chromosome number during meiosis I. The functions of MOKIRs have been studied in 4 different organisms: 2 yeasts, the fruit fly Drosophila melanogaster, and mice. However, the makeup of these proteins, in terms of their amino acid building blocks, is actually quite different, and yet they seem to perform very similar functions. One of the key roles of MOKIRs is to make sure that sister chromatids are segregated to the same daughter cell in meiosis I, rather than being separated prematurely. Early in meiosis I, homologous chromosomes become connected and exchange genetic material before being set up for the first meiotic division. Research from many labs has shown that MOKIRs help the co-segregation of sister chromatids by regulating a kinase called polo. In budding yeast, fission yeast, and mice, the critical function of MOKIRs appears to be to recruit polo kinase to a specific region of the chromosome called the centromere. There, polo presumably modifies other proteins with phosphate residues to ensure the co-segregation of sister chromatids in meiosis I in ways that are not yet understood. In the fruit fly Drosophila, the respective MOKIR instead appears to act by preventing polo from localizing to chromosomes, although it is currently not clear whether and how this promotes sister chromatid co-segregation. The second conserved function of MOKIRs is to prevent the premature separation of sister chromatids in meiosis. Sister chromatids are typically cohered and prevented from premature separation by a protein complex called cohesin. Cohesin is a ring-shaped molecule that enwraps sister chromatids and thereby holds them together. During a distinct phase of the cell division process, an enzyme called separase can make a cut in the cohesin ring, thus causing sister chromatids to separate. In the early stages of meiosis, homologous chromosomes are entangled and linked together at regions away from their centromere. Later in meiosis 1, cohesin cleavage everywhere except centromeres releases this entanglement and causes homologous chromosomes to segregate. However, cohesin at centromeres is protected from cleavage until meiosis 2. Then, separase becomes active once more and cleaves the remaining cohesin to allow the separation of sister chromatids. Along the length of the chromosome, cohesin is modified with phosphate groups by cohesin kinases, and this promotes cohesin cleavage by separase, as these phosphate groups make cohesin a better target for separase. To protect cohesin around the centromere, a protein called shigoshin ensures the removal of phosphate groups from cohesin in this area of the chromosome. It is this removal of these phosphate groups that prevents separase from recognizing and cleaving cohesin at the centromeres. Evidence from the literature indicates that MOKIRs also help cohesin protection, and that they do this by two mechanisms. Firstly, we think that MOKIRs somehow suppress or restrict the activity of the kinases that act on cohesin, thus reducing the number of phosphate groups that these kinases can put on cohesin. And secondly, we think that MOKIRs ensure that shigoshin can act to remove phosphate groups from cohesin until meiosis 2. However, the mechanisms by which MOKIRs safeguard homologous chromosome segregation and cohesin protection during meiosis 1 still remain elusive. In order to figure out how these proteins work, we need a better understanding of the ways in which the first meiotic division differs from mitosis. Addressing this question is also important for human health, because errors in meiotic chromosome segregation can cause infertility and leads to developmental disorders such as Down syndrome.